All right, I think we're live. Hello, everybody. So this is a first for me. I've done presentations and tutorials and webinars and things like that, but I've never done a live stream before. So this will be a bit of an experiment. Hopefully not too much of a bumpy ride, but we'll see how it goes. I feel like I've got my battle station all sort of set up and configured here, but I hope to be able to do more live streams in the future. So if this one goes okay, then I'll improve and keep learning and stuff like that. So yeah, it's this will be interesting. Welcome to the people that are here already. I see there's a few people in the chat. Great to see you. And I think the reason I wanted to try live streaming right now, obviously there's a lot of what with the pandemic going on, there's a lot of people at home more often than not, and maybe I can offer a little bit of entertainment, a little bit of something interesting to watch and divert your attention. I've been meaning to do some live streaming for a while, but this is now seems to be as good a time as any. So yeah, if you notice any sort of technical issues or if the volume levels are way out of whack or anything like that, do let me know and I'll try to fix things on the fly if we run into issues. But yeah, in the meantime, so this is going to be a casual setting. I'm not doing a formal tutorial or anything like that. So if you're hoping for in-depth instruction, I don't know. Maybe you'll get that. Maybe you won't. I'm not sure. I'm just going to be working on a shot from an upcoming animation, and I'll try to explain my thought process as I go. But I don't know. Maybe I'll go in depth. Maybe I won't. I do like to ramble, as you can tell. So I'll probably be talking the whole time and explaining every little nuance of everything. So tell me if I'm doing too much of that or not enough of that. And feel free to ask questions in the chat. I'd love to get your thoughts on what you're interested in seeing, and or give advice. So for those that don't know me, I'm what you call a generalist, which means I know some about a lot of different things: biology, biochemistry, computer animation, graphic design, programming, mathematics, and you'll probably see a little bit of all of that today, but I'm not an expert at any of those. So if you are an expert and in whatever it is I'm doing at the moment, feel free to give me some pointers and suggestions and stuff like that. So on that note, I don't know it looks like I've got about 15 people at the moment, which is awesome. That's more than I was expecting at the beginning. So we'll see if that number goes up or goes down over time. And if you have to go or you come and go as you please, that's totally cool. This is a informal setting. So yeah, given that I don't know exactly the demographic of people that are watching, if you would like to introduce yourself in the chat, not necessarily your name if you don't want to give your name or anything like that, but if you just want to say like what your connection is to me or to the work that I do, or what you're most interested in, if you're a medical animator, or if you're a student that likes watching science videos, or if you're a friend or family member or whatever. I'd love to see who's kind of checking out the stream at the moment. Yeah, so I think that's enough preamble. Let's get to the actual doing stuff, because that's probably more interesting than a talking head. So I'm just gonna rearrange my workspace a little bit, and I'm probably gonna, even though I've kind of got this set up a little bit how I want to, I'm probably gonna have to do some window juggling and everything like that. So forgive me if I'm having to blank out the screen and shuffle all my stuff around, because I've got way too many things open right now. Okay, Houdini. All right, here we go. So today, a little bit of sort of context for what I'm doing. So I'm making an animation about molecules. I'm not going to tell you exactly everything that's involved, but one thing that I'm going to be doing is showing amino acids in the video. So what I wanted to do today was maybe have a layout where I can see all 20 amino acids, and then I'm going to be doing a little bit of cool animation with them in later on. So that's the plan. And I was really resisting the urge to figure everything out ahead of time so that this is more like a presentation. But this isn't a cooking show. I'm not going to be doing like, here's one I prepared earlier that's already done, you can check it out. This isn't a cooking show. So I'm going to be doing things on the fly. I'm going to be figuring out what I need to do as I go. And so that part might be a little bit of a struggle, but it's also more realistic to my work process. It's more like actually a lot of problem solving and stuff like that. So hopefully we'll get to see some problem solving. Yeah. So question in the chat, can you post the whole live stream afterwards? Yes. I think by default, the YouTube captures the whole live stream and it becomes a whole standalone video. So it should be able to be viewable on the Making Biocinematics channel, which is the channel that's streaming right now. So if you're not subscribed to that, feel free to do that, or just come back to the same link. I think it should be the same link if you wanted to rewatch this after the fact. Yeah. Unless something terrible goes wrong, in which case I might have to pull down the whole thing. But barring that situation, yes, this whole thing should be available afterwards. And maybe that's what you're watching right now. Hello from the past. Okay, so step one. Hey, lots of people I recognize in the chat. That's really great to see. All right, so I shouldn't leave you too long on a blank screen here. I should actually do something, shouldn't I? All right, well, so I'm not starting from a blank scene, and I considered starting from a blank scene in Houdini because that would be more of a good beginning, but that wouldn't be realistic. So I'm basically starting with a template scene that I use for a variety of shots in the animations. Maybe what I'll do is I'll pull up the trailer just so you have an idea of the kind of final thing that I'm sort of looking for. So let me see if I can pull up. Oh man, hello. Lots of people I recognize. That's fun to see. How are y'all doing? I hope everybody is staying safe and comfortable and able to be, yeah, carrying on life in a slightly different format, but yeah, I'm getting a little bit of an echo from my feedback over here, or I've got an iPad that's playing the stream so that I can actually see what I'm doing. There we go. Okay, so so this is an example. So I posted this trailer in December for the video that I'm working on right now. And the reason, beyond the fact that this is a far too long and ambitious project for what I actually should be doing, the reason that the video isn't out yet is I've been doing a lot of client work in the past few months. So I posted the trailer and I was like, it's going to be out in early 2020. I guess technically this is still early 2020. So by the time the video actually comes out, we can decide whether or not this is early 2020. But yeah, that was, at least I didn't say January. That would have been embarrassing. Okay, so what I'm going to be doing is I'm going to be making some molecules in this kind of style. And so I've got a lot of these shots sort of have that format. We're showing molecules against this backdrop and sort of talking about them. So let's do that. So as I mentioned, I'm going to be trying to display all the 20 amino acids in the scene. And the way that I want to do that, what I could do is I could search. So it's hard to know how much detail to go into, but I'll go into a little bit of detail. So when I'm modeling molecules, what I want to do is I want to find structural data. And the best place to do that, as many of you know, is the protein data bank. So what I could do, let's see if I can grab. Actually, what I'm going to do is just search Man searches for something on the internet without the internet being on screen. I know it makes for riveting viewing. All right. So here we go. Okay, so what I could do to get these amino acids, so as a primer on the biochemistry, there are 20 different kinds of amino acids that humans and pretty much all organisms make use of. And so valine is one example. So stop rotating. Okay, there we go. So what I could do is I could grab the files. There was data files associated with each of these and bring them into Houdini one by one. But what I kind of want to do is not have to do that 20 times because that would be a little bit tedious to do. So what I'm going to try and do is do it a little more procedurally, so I can grab all of the amino acids at one time and work with them in parallel as opposed to doing them individually one by one. Yeah, so maybe to start, what I'll do is bring in this valine, and then I can think of a way to bring in them all together, so I know that I have that at least as a starting point. So I'm going to download. Actually, I'm not sure where this is going to save this. In case this saves it into a confidential client folder, I'm going to jump back to my webcam here. For the time being, I might have to do that a little bit back and forth, and I apologize for that. But it's not like I have anything that secret, but you know, it's just how I feel. Oh, it did go into a client folder. Yeah, you can't see that. That research hasn't been published yet. Okay. So I'll go into here, valine. Now, did I grab the ideal or did oh, okay. Sometimes on those ligand pages, there's an ideal structure and a model structure, and I'm not sure exactly the difference. I'm assuming that the ideal is some kind of like mathematically, here are the atoms in this position, and that's like the arrangement that they'd be in a more ideal scenario, and then a model would be something that's derived experimentally, but I'm not sure if that's actually true. I shouldn't make up stuff. Okay, whatever, valine. All right. Yeah, so just to show that, so you can download, so what I download is the CIF format, and then I'm gonna have to convert that using Chimera or something like that and bring it in, or the SDF. I think that, I can't remember if I can use that. Okay. Now I forgot that I was gonna have to do Chimera, so I'm gonna have to, again, juggle my windows here. Hello Nick, hello Ruth, hello Kevin, hello Susan, hello Lucas, hello Genevieve, hello Mansan, everybody. That's great. All right. Sorry, yeah, I have to. I should name client projects in a really, really cryptic way instead of something a little bit more obvious, and that way I wouldn't have to hide so much stuff. Oh, come on. Okay. Plan B. So the other tool so Chimera wasn't working, wasn't either responding or wasn't reading the CIF file, if somebody's like, no, 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 you can't use Chimera for CIF data files, let me know what I should be doing. In the meantime, I'm going to use a tool called Mercury, which also is good for doing crystal structure conversion. But the CIF file didn't work in either of those, so I think I'm going to try the SDF. That's probably what I did last time. And last time, I mean every time. But like I said, it's been a little while since I've gone into this project. So a question, when did you start working with Houdini and how long did it take me to learn? The learning curve is steep. The learning curve is steep. If you have experience with other 3D software, that is very helpful, but at the same time, it's very much a different paradigm. It's not like going from Maya to Cinema 4D or Blender to Maya, or Maya to Max or something like that. We'll see that a little bit as I work, but yeah, it's quite different. So I started learning. So here we go. So okay, so I've got the Valine into Chimera now, and all I'm going to do is I'm going to export it as a PDB, protein data bank file, which is what Houdini can read natively, which is one of the reasons why Houdini is so awesome. And I really like it because you can work with the PDB data very directly in a very sort of hands-on way. So I started learning Houdini last January, so it's been a little over a year. And I'm not an expert. Like I still feel very much like an amateur, so that'll be fun to see. But yeah, it does take time, and it's a very different way of thinking about making through computer animation. But as somebody who's dabbled with programming a bit and coding, that it's more of that kind of thinking. So if you have sort of like on the opposite ends of the spectrum, you've got a very sculptural approach in ZBrush, for example, where it's just like you're directly manipulating something. You're using your artist brush, your artist hand to create something. In Houdini, it's very much of almost like you're creating an algorithm to generate something or generate many things, which is where it's really handy. Hey Sam, how you doing? Good to see you. So yeah, so I'm gonna save. For most of this, I'm not gonna be like saving and loading files. Just at the beginning here, I have to save files, which is why it's a little bit back and forth with the webcam. But you can see my face bigger here, so that's always a bonus. All right, let's jump back in here. All right, okay. I think this should be okay. Okay, so now that I've saved this out, so I've created a little PDB tool for myself, which is basically a way that I can just generate different molecular representations quickly. So I said this is not like a tutorial, like I'm going to give you the ins and outs because there's a lot of secret sauce in here and I can't share all of that. Actually, it's not that secret. It's just, yeah. So I'm going to grab my data, PDB, Valine, and do we have any data here? We do not. That's because I need to include head atoms. There we go. Is that there? Yeah, that's there. All right. Are there hydrogens? There should be hydrogens. There are hydrogens. That's great. Okay, so this is our Valine. So this is going to be my starting point for generating the rest of the amino acids. And what I kind of want to do is use a ball and stick representation. So if we enable ball and stick, you can't actually see anything because the atoms are too big. So I think I go to 0.4. I can't actually remember what scale I'm using. I should check that, double check that before I bake anything. But the nice thing about this being procedural is it absolutely doesn't matter what settings you choose at the beginning because you can always change them later. And I can color the sticks if I want. I'm just showing off some of the options in my nice little PDB tool. Yep. Okay, so I think all right, audience interaction at this point. So what I want to do to grab all of the other amino acids is I want to grab one protein that has all 20 amino acids. What protein do you think has all of, at least one of each amino acid in it? Do you think that's common? Do you think that's like something that's frequently observed? Because there's, in the average protein, there's probably several hundred amino acids, but you're probably going to have the same common ones most often, right? Like, I don't know, glycine or whatever's in the alpha helices. So I don't, this isn't like a quiz. This is like, I actually don't have any idea. And I was going to look this up beforehand and I was like, no, this isn't a cooking show. Hemoglobin. Okay. You know, I actually think I have hemoglobin as a, we can check. So that's a good suggestion, at least as a starting point, because then we can check, and then if it doesn't work, then we can look for a bigger protein, and presumably the bigger protein, the bigger you go with proteins, the more amino acids you're going to capture. An egg has all of the amino acids you need. So in egg, I think isn't it albumin? Like albumin is the common protein in eggs. That's a good option. We could look at that one next if hemoglobin doesn't have all of them. Or I could Google it. I wonder if maybe somebody can Google if there's an individual protein molecule that has all amino acids. What am I looking for? PDB. Albumin, the white in the yolk. Yeah, that's a good point. I mean, there's probably lots of different types of protein molecules in an egg that maybe together contribute to kind of having everything. Okay, so I don't have hemoglobin in this folder, so I think I know where it is, but I'm just gonna have to do my there we go, dance here and just look for it. Actin is a pretty small molecule, but in terms of like protein, I think, but that's also one I actually, you know what, I might have that one already right here, because I think I used actin in okay, so I got hemoglobin here and actin. Okay, so we could try both of these. So let's do this one. I'll pull up the camera in just a second. All right. So this one, there's actin. So how many amino acids does it have? All right, so for those that are, oh, you checked. Okay, cool. Well, then let's let's go for that. So can you see in the bottom? Yeah, you can see in the bottom here. So this is the Houdini. So we've got the 3D viewport, which you can kind of see the end result of what you're trying to make. But then the spreadsheet with a bunch of data, which is more nerdy, but it's more useful when you're trying to work with the data directly. So what we have here, actually, if you want an introduction to molecular visualization with Houdini, the Entagma just did a recent collaboration with, I think it's Dr. Jeroen Klaus, who, here we go, this here guest tutorial. So this is rendering the coronavirus spike protein, and he actually goes into more granular detail about actually pulling in the, yeah, somebody just in the chat. Yeah. That's the one that, yeah, he goes into detail about like what it means, why it's important to have that structural data, and how you can bring it in and do stuff with it. So I'm going to be doing slightly different stuff than in terms of like the visual representation today, but that's good background if you haven't taken a look at that. And this nerd chat, as they call it, an interview with him, is also really informative about kind of the thought process and coronavirus. And yeah, I know that some people, like Nick, did a great coronavirus model that, oh, what if I'm just showing stuff, I might as well just grab. So if now that we're talking about coronavirus stuff, so the Master of Science in Biomedical Communications, which is where I did my master's, and a lot of the people that are watching right now have also gone through that program. They've got a page here, which is really cool, the COVID-19 visual media. So you can, like, there's some cool resources. And that's another example of using, I think, Nick, we're using Cinema 4D, I guess, for this visualization. So Houdini is not the only program that you can use for molecular visualization, obviously, if that's something you're interested in. All right, let's get rid of that. I don't know why did I start talking about that? Oh yeah, data. Okay. We'll see how long my throat holds out too. This is probably longer than I've talked in months and months already, working at home. Okay, so we've got position data, we've got color data, which I've added based on the element type. So we've got the element, we've got here, we've got the residue name. So this is what I'm interested in. This is, so we can see that the first one, and they're starting here, sequence 4, or the sequence index 4, is cheat sheet. No, I don't even have my cheat. Oh, I do have my cheat sheet. Is that glutamate? It is glutamate. I knew that right off the top of my head. And then threonine, alanine, leucine, valine. So here we go. So we've got valine here. So we can compare this with our one that we pulled from the protein data bank before and see if they're the same structure. I hope they are. Cysteine, aspartate. Okay, so yeah. So what I think I might do is try to just iterate over each of these. And I'm going to do that with an iterating for loop to go over the named primitive. No, I want to go over points. And I'm going to call iterate over the name. No, that's not what I want to do. I want to iterate over the res name. So I'm just choosing the attribute that I want to iterate over. Kevin asked the question, speaking of the virus, there's so many medical illustrations about the virus. How do you determine which of them are scientific accurate enough and which of them are more like art? That's a really good question. I'm probably not the best person to speak to it, but since I'm the only person speaking audibly in this room, then I might as well try to take a stab at it. There's obviously, if you've been on the internet in the last couple of months, there's an explosion of depictions of coronavirus stuff. Some of them are clearly more scientifically accurate. Others are more artistic representations. And there's value in both. I mean, most of the time, not to be not to be sort of diminishing the importance of it, but most of the time it seems like they're being used as sort of an editorial, like, here's an article about coronavirus, let's include a picture of something that looks like a coronavirus. And other times it's being used more as in a more educational capacity, where you're talking about things like the proteins on the outside that bind to host receptors and stuff like that. And so I think there's a slight so if you have an editorial illustration, is it necessary for it to be completely scientifically accurate, and how far do you go? And even with the ones that are scientifically accurate, it's not 100% accurate because we don't have all of the data. And even if we did have all of the data, it's almost impossible to pack all of that data into a single illustration sort of thing. So I think for the lay public, i.e. most of the people that are looking at these images, I think it's almost impossible to tell which ones are more accurate than other, because that, like, they don't have so much to go on. If somebody says this is a picture of a coronavirus, then a lot of people think this is like somebody used a microscope and this is an actual coronavirus that we're looking at, when in fact it's a sphere with some extrusions on it. So I think that that's where we can have, in sort of like a medical illustrator, scientific animator sort of field and profession, we can kind of lift up the ones that are using real data, using informed approaches to making images, and kind of using that as a way to educate the public about this image is valuable because of the work that went into it. I think somebody like David Goodsell, who's got a lot of aside from the amazing artwork, there's a lot of data behind it, and so there's this kind of that beautiful blend of something that's aesthetic, something that's captivating to pretty much everybody, in my opinion, but also has a lot of data behind it. And you can kind of use things like that as models of, like, this is artistically excellent and also carries a lot of useful information behind it. I don't really know. Yeah. So that's that. Maybe I just talked around in a circle. I don't know. The answer is most people can't. So the people that can kind of boost the stuff that they think is valuable. All right. So let's see here. So it looks like, what do I want to do here? So let's put down a null. A null is just sort of like an ending point, some marker where I can say that I want to look at a result. So by here, it looks like gather 20. It looks like it's grabbing each individual amino acid. But what I think I want to do is I'm going to try a couple things without really explaining too much. And then maybe at the end, I'll sort of go over my approach, because I don't know if this is going to work. I'm just going to be experimenting a little bit. So I'm going to talk a bit less for a while. I'm also going to have a fisherman's friend to help my throat, because I figured my throat wouldn't last too long, but this is what training is about. If I'm going to be doing more live streams, I need to train myself to talk a lot. All right. Yeah, so let's do. Oh. I did something. What did I do? Oh, I know what I did. So I'm in the first pass. So all I'm doing is I'm grabbing, yeah. So these are all of the alanine residues. And that's interesting because it looks like, I don't know, alanine is a small, yeah, alanine just has a methyl group on there. So assuming that, let's do this. One of the really useful, one of the reasons I like ball and stick representation is when you're looking at a small molecule, the difference between trying to get your head around a structure that looks like that and get your head around the sort of the spatial sort of arrangement of atoms and something that looks like that is very different. And this is where it's really handy to have a ball and stick representation. So this looks like a complete alanine residue without the hydrogens, and hydrogens is something I'm going to have to tackle later. Now, if I was working in a different application, I'd have to make sure that I have all of that up front, but because I'm working in Houdini, I don't have to worry about that until later, and we'll see if I'm telling the truth or not. That might be a horrible mistake. But if I set this up properly, I should be able to pull in any protein structure and check if it has all the amino acids and set them up accordingly. So what do I want? So I've got all of the alanine residues here. I just want the first one. I just want one of them. So what I'm going to do is use a wrangle. And a wrangle is a little bit of code. So now I get to use code. Question about more molecular Maya making of videos. So I'm pretty much using Houdini exclusively. So I'm not using molecular Maya as much, which is part of the reason I'm have to kind of make my own PDB molecular representation tools. But I think Maya is really good for a lot of things. So at some point I might sort of return to that and do more molecular Maya. If you're asking more generally about like how-to tutorials outside of like in Houdini, for example, yeah, hopefully I'll be able to do more tutorials. They might be more, I imagine I'll be doing more sort of behind the scenes stuff in addition to these live streams about various topics and kind of take a more high level than a strictly sort of tutorial step-by-step approach. But I might be doing some tutorials as well. If you have suggestions of specific topics that you'd like to do, like to have tutorials on, please let me know via send me a Twitter message or Instagram or Facebook or email or whatever. Okay, so what am I doing? I want to isolate first amino acid. So the first amino acid basically I want to look for the smallest res seq. That's what I want. I want to find the minimum. So what I could do, this is where it's going to be a little trial and error. I'm going to do a detail, and I'm going to this is where I need to grab get the documentation. No, you know what? It's find attrib_val. I mean, I could do this in a sort of. Oh, I know what. I know. Let's forget that. Forget that. Okay. Lowest seek res. And I don't have to choose the smallest or the first one in the chain, but why not? So I'm not going to explain all of the Vex. So this is Vex. This is Houdini coding language. But this should go away there if I don't have this is where it's very handy to have a little bit of coding experience, but a little bit is also dangerous because you end up turning to that for solutions and then they get really complicated really quickly. But basically what I want to say here is we're going to loop over all of the points that are currently in the total group of alanine points and try to check if they are part of the amino acid that, no, I already got it, that I want. And that's not what I want. I want the current point number, then all right, so the lowest debugging in Houdini is kind of weird, and it's not something that I've been good at. So when I have more time, then I just do a bunch of trial and error, sort of like hack away at it. But when I'm trying to be all elegant and sophisticated in front of a live audience, then I feel like debugging would be something that would be really handy. Especially because I don't use this very often. Zero. So what I was looking for obviously was 7. Oh my goodness. Res seek res. Why couldn't they have called it something more memorable? That is short for residue sequence. So or something like that. It's basically the index that individual amino acid is in the whole protein chain. So look at that. We've isolated a single amino acid. Huzzah. All right, now if I turn off single pass, and I get rid of my print statement because I don't like the console popping up. That looks like a whole bunch of different amino acids to me. That's successful. All right. So how do we want to check this? It says gather 20, which means that there should be where's 20 unique. There you go. Yeah, 20 unique identifiers for the residue name, and the residue name is the 3-letter acronym for the individual amino acid. So we've got alanine, arginine, asparagine, aspartate, cysteine, glutamine, glutamate, glycine, histidine. That's what it's called, right? Yeah. Histidine. All right, so now we've got these amino acids. Let's save that. That's cool. But all right, actually, maybe I'll sort of explain this a little bit better, because that understandably was some incredible wizardry that nobody could figure out. I mean, you gotta love Houdini, because like it's at a point where it's like it's not working, and then all of a sudden it works, and you're almost as surprised as anybody who's like not doing it themselves. So I've got my original protein chain here. And what I want to do is I'm going to iterate on a collections of atoms. So each collection of atom is going to have the same residue name. So first we're going to iterate on all of the alanine residues, and then we're going to iterate on all of the, whatever the next one was, arginine residues, etc., and do that for every single one that there is. And then what I wanted to do was I wanted to just grab the first amino acid in that collection. But and to do that, basically I'm checking, do these all have the same sequence identifier, which means that they are part of the same amino acid. And then blast, which is basically a fancy way of saying get rid of it right now, gets rid of all of the other ones, delete non-selected, right? So I'm checking for my first amino acid group and then deleting all of them. Then I'm running that, because what that does is it creates individual points, a whole bunch of little points. Then I pipe that back into my secret sauce PDB structural thingamajig. And there we go. Now we've got all our different amino acids. Now the next part is going to involve some mathematics, because what I want to do is I want to orient them. Actually, you know what? Before we do that, let's solve the hydrogen problem, because this actin molecule. So in the protein data bank, pretty much all of the proteins are stored without hydrogen positions, and I'm trying to remember if that was because just basically like storage capacity. It's simpler to ignore those, and in most cases you don't need them, or if it's because experimentally it's hard to determine the actual positions of the hydrogens. Excuse me. But in any case, fortunately, there's a web tool that can do this for us. Add the hydrogens sort of in a computational way. So that's what this molprobity server does. So what I'm going to do is I'm just going to put in the PDB ID, and that's the actin that I was using, I think. I hope. What? Well, all right, uncomplexed actin. Cool. No explicit hydrogen atoms are included. That's what I suspected. So now we click on the button that says add hydrogens. And do I want to flip so these in, doesn't matter, too much detail. Let's carry on. So what, Is everybody still good? Oh. In my YouTube analytics, it says like it dropped down to 0 concurrent users or viewers, and I was like, uh-oh, that's no good. But it seems like we're all good. Does anybody want to say something and make? I think it's still all good. But anyways. We're good. Okay. People are still here. All right. Do I want to flip? Sure, whatever. So basically flipping is like if you've got certain amino acid residues have kind of like a fork structure piece. And whether it's the atoms are this way or this way, it's very, very difficult to tell. So from an X-ray crystallographic standpoint, like they just make a guess, I think. And then in the computational side, when they're adding the hydrogens, they say, oh, hang on, there's actually more evidence to support this conformation instead of this conformation. So they kind of like, do you want to flip the stuff for you? So it doesn't, for our purposes, it really doesn't matter, but whatever, why not? What? So added H, yeah, okay. When they have a big red warning across the top, it's a little concerning. It's like, what happened here? Okay, so I'm gonna switch back to my camera. There we go. And download the file. Flips. Question, couldn't use preamble to add the hydrogen, correct the structure, etc. What's the advantage of scripting in Houdini using mole probability? That's an absolutely fair question. I didn't know that preamble had that functionality built in. Maybe it's using the same server or the same kind of mechanism as this. Scripting in Houdini is going to be for separate things. So, I mean, maybe there's a way in preamble to do what I'm trying to do here. So the adding the hydrogens is independent of Houdini. I'm just going to do that, and I'm just going to re-import the new file that has the hydrogens versus the one that doesn't. So that's the only reason. And this was the simplest way that when I was searching for how to do this, the simplest way that I found to add hydrogens. But if preamble can do that too, then that's awesome. PDB file with hydrogens. There we go. I don't know why they make it so hard to figure out which file you actually need to download. But that's like a very minor problem. The fact that I can add hydrogens for me is super awesome. You know what? Actin with hydrogens. Okay, I'm just gonna, again, just grab the path so that I'm not mucking around with too many file windows. Here we are. All right, so we're sort of at the beginning of our chain here. Let's clean this up a little bit. Sorry, I didn't do the hemoglobin route because it turned out that actin was a decent option. It's cool that even a relatively small structure can have quite a lot of, like have all of the atoms, in which case I'm assuming that hemoglobin, which I think is much larger structure, would also have all of the amino acids. Okay, so basically here I can switch between the two. So without hydrogens, and with the hydrogens, Now, the reason that I'm having, so you'll notice a couple of differences. One of the differences is that we are losing every other atom. Chimera can add H as well. Oh, cool. I didn't know that. Here I was going the long route because I realized that what I actually have to do now is run a Python script to fix that. Because what happens with the multiprobability serve is when they're generating the PDB file, they muck with the line endings. And the resolution of the video is dropping a bit. Hopefully the stream quality is still okay. Nothing's changed on my end except for maybe the fact that the ISP is like, wow, this guy's uploading a lot of data right all in one go. Hopefully that's not an issue. So multiprobability is, yeah, mucks with the line endings, in which case Houdini doesn't read the PDB files well, and I forgot about that. So I've got a couple options. One, I can try to do it in Chimera. I don't think I have Pymol installed right now. And muck through that, or I can just try to run the run my little Python script. I think I might try to do the latter. If you want to I'm going to go off screen for this little bit, partly because I have no idea what I'm doing, and partly because here we go. Okay. If anybody wants to take a bathroom break or anything like that. So we've been going for almost an hour. I think I do want to carry on. I think this is, hopefully from your perspective, this is going okay. I think it's going okay from my perspective, and I'd like to try to get a bit further with the actual arrangement of the amino acids. Everything always takes longer than you expect it to, especially when I talk for so much time. It's not actually that different from a regular work day for myself because I talk to myself a lot too. But anyways. All right. So what I'm doing behind the scenes, I'll give you a blow by blow. So what I'm going to do, because I haven't got my Python path set up properly, because I haven't, is I'm going to grab my PDB file that has the actins, and I'm going to put it in a new, I'm going to put it in the same folder as my Python script, if I can remember where that is. There we go. And then I'm going to run fix reduced pdb.py. Input file is test files. That's right, I've got a folder called test files, specifically for production files. It worked. Now I've got my fixed file. I'm going to paste it back into my production folder. And I'm going to jump back to Houdini. Ta-da! Wait. Ta-da! It was my idea to add a little underscore fix to the file. That's it. Yeah, it's akin to like final, adding final to files. Okay, so now we've got hydrogens on all of our amino acids. So that was roundabout. And ooh. That's an interesting, okay, so what we have here is we've got a whole bunch of amino acids linked together. And I want them individual, but I don't think that should be too much of a problem, because what we're going to be doing next is basically adding the bonds and the atoms is the last step in the process. So what we have before that is just points in space. And in terms of points in space, it doesn't really matter what the atoms are connected to, because the bonds are added as kind of like a post hoc operation. So we're fine there. But I suppose it makes sense that a lot of them would be linked together, because those are the first instances of each of those type of amino acids. All right, so as I was saying, we've got amino acids with hydrogens, and what do we want to do next? I want to lay them out. Do I want to do anything else first? I had some notes of what I was going to do. I mean, this is all on the fly. I didn't prepare anything. Yeah, orienting them consistently. So first, what I want to do is I want them all to have this similar orientation, so that you can see the differences in the R groups more easily. And the next thing that I want to do is once they're oriented properly, I want to spread them out on a grid, or similar to a grid. We'll see what happens. Yeah. So orienting them consistently. So Chimera doesn't do the flipping. I honestly like adding the hydrogens is really all I'm concerned about for that kind of operation. But flipping is kind of I guess, yeah, I don't really know. For my animations, I don't think the flipping is going to make too much of a difference. I don't think any viewer is going to be like, hang on, that histidine side chain doesn't look to be in the proper orientation, especially when they're independently. There's going to have, anyway, yeah, freedom of motion. Anyways, so the orientation is where I'm going to have to do some math, and this is where Houdini gets a little hairy, because math is, like I said, I'm a generalist. I know a little bit of math. I don't know a lot of math. And this is where you really need a textbook on 3D computer graphics mathematics, which I may have. Two, in fact, I've got 2 textbooks on math for computer graphics. And I've read the first 6 pages of each of them. So I'll get there one day. So what we want to do is so this is what I'm thinking. Let's first orient the valine as our reference, so that we have kind of a baseline for what we want to do. I'm going to hide this, the other stuff in the scene, so we're not distracted by it. So what I want to do is I'll put a transform node, which allows me to transform the thing that I've got. And I think I think what I want is the R group down, right? So let's put this, we want this at the origin to start, because we're going to be spreading them out in a separate step. So what I could do, yes, I have this functionality built in, move atom to origin. So atom to move to origin. Wait, why is that? Oh, because of transform. Yeah, wow. Okay, so I had some extra transforms at the object level. So for those of you not familiar with Houdini, you've got sort of, this is the object level. So I've got this sort of container of geometry, and I had some translates and rotates on it. So it was like moved in space, so that when I was working at it at the SOP level, the geometry level, the individual vertices, points, and stuff like that, it was offset. So this is going to give me a better, a more accurate look at what I'm actually doing. So I want to move the alpha carbon to the origin. I think that makes the most sense. Is that the alpha carbon? It seems to be. So the alpha carbon is snapped right to the origin, and now we're going to rotate this manually so that the, you know what, we could do, and I could do this, I could actually do this more exactly if I was doing it in sort of a mathematically perfect sense, but I think that'll do for now. And then in terms of the top view, I think let's try to orient it sort of along that plane, so that the carboxyl group and the amine group are yeah, sure, that'll work for now. So they're kind of like in a line. One thing that in the when you see sort of one of the downsides of a 3D representation of a small molecule is that very often you have, there's not a single view that gives you all of the information that you're trying to convey. So the idea that I'm trying to put all of these amino acids in kind of like one view, maybe I'm doomed before I begin. But I'm not necessarily trying to teach in this particular shot about like the individual nuances of each individual amino acid. I kind of just want to get across the idea that there's 20 different flavors of this particular molecule. So that'll do us for now. Okay, so that's kind of our reference orientation for I'm just trying to think if there's a better, because what I'm planning is doing kind of setting the position of the alpha carbon, and then kind of trying to match the orientation of the carboxyl carbon and the nitrogen. I should get around to learning PyMol someday. I'll add it to the very long list of things that I should get around to learning. In the meantime, I'll just stumble along and do things as I go, bits and pieces. All right, so let's do, again, we're gonna do another for each named point, because we're gonna do this on each individual amino acid at a time. What was that? That was nothing. Could we do this all in one step? Yeah, I think so. Okay. So we're still using the same main loop. Now we're going to, let's break this up. We're gonna place at origin, and then we're going to orient so good. I make it a habit to name all of my nodes sensibly. Right is ambiguous. I should use something more unambiguous. All right, place at origin. So I guess I'm just gonna do that. Another really nice thing about Houdini is you frequently don't have to the pivot point. Actually, yeah. What was I saying? Repeating yourself. Ironically, I was talking about repeating myself. You can just grab work that you've already done, paste it in, and that really gets you a lot of the way forward. The downside of that is I feel like I don't learn things as well when I'm not forced to do them from scratch over and over. I just kind of learn how to do it once and then just kind of copy myself doing it over and over. But that's just the nature of trying to make things happen. So what I'm wanting to do is I'm wanting to, why is that? Set the pivot point to be the location of the alpha carbon. So in fact, I'm gonna just rearrange these. I'm gonna call this set alpha carbon pivot. All right, and so presumably for each of these, now we're at, I'm just gonna do this. Let's do a single pass of this and just worry about the alanine for now. So what I want to do is grab the alpha carbon, and the alpha carbon has a very specific, let's tag this with the name. I think that's what the attribute is called. Yeah, name. So the name has a little bit more information than just the element. So we've got nitrogen, obviously. I think Ca is the alpha carbon, but I want to double check that. And I think that one is the carboxyl carbon. So what I'm going to do is, tag it with the, there we go. So now I can see that that specific name attribute on each of the points. So Ca is the alpha carbon, and so that's what I want to do. So I'm going to grab the I can't remember the syntax for setting the vector type. We'll figure it out. So we're going to get the position in world space of the first data stream. That means we've got this little input here. The position, that's P, of, first we got to get the alpha point is find attribute val name is what it is, Ca. There we go. And then we want to find what? That's when you got the documentation, which is only F1 away. So attribute class, that's right. There we go. Can you hear an echo? Because I can hear myself through the headphones that I have here beside the iPad, which I'm using to check that the live stream is still up and working. Hopefully that there's no echo. Okay, so yeah, we're doing the mathy part, so this part may be a little bit dry, but hey, yeah, that's what it is. You guys tuned in to watch me. It's not my fault if what I'm doing ends up being vector maths. That's the field that we're in right now. Vector maths. All right. So I think we need to have this as a detail attribute so that we get the pivot point. There we go. Okay, so we're getting the wrong pivot point. What are we wanting to do? So see, I just did this a moment ago, so now I know. -1. Okay, so that means what it's doing is it cannot find this attribute val. Right, because now that this is a detail, it's not necessarily understanding that this is a point attribute. So is there a function? Oh. Attribute class. Not string. Point. There we go. My molecule disappeared. Why did it disappear? Because it's now at the origin. Hurrah. Okay. So now what we've got is if we do this for all of them, we've got a whole mess of stuff here. I had to turn off this. Yeah, well. Ignore the sticks because, yeah, they're all going to be, it's like a spider web. You can see how easy it is to create some interesting things if you're not worried about doing something scientifically relevant. You can make some pretty neat art just mucking around in Houdini, which is something I'd like to do someday. Oh, every time I stretch, the top of my head gets cut off. That's the problem with having a green screen setup, is it's unnatural to see parts of somebody's body just kind of disappear. Whereas, I don't know. But I figured that having the green screen was better than just a blank background, because now you can see, if I duck, the detail attributes here. Or I can just jump over here, but then I cut off my node network. That's too good to cut off. All right. I should... all right, so we've got the placing at the origin. That's good. Now we need to orient the correct way. All right, so we're going to do single pass, and we're going to do take this and use this one as a reference. So you can see our reference, if we just... Sometimes the viewport kind of gets stuck, and I haven't found a way to kind of refresh it. Because what I believe is happening right now is we've got... Oh, there we go. Never mind. So we've got a reference valine molecule here. And what I want to do is match this one to that one. So why is... It looks like the alpha carbon's slightly offset right now, so that's what I want to figure out first. Yeah, what's going on here? Oh, is the I just want to make sure that before I get to started at like doing, yeah, okay. So those one, that's lined up now. So it seems like when we do that, the rotation, this gets bumped up. And I feel like that's because the pivot, my pivot isn't 0. How did that happen? Okay. So there we go. Okay, so now we got the alpha carbons are in exactly the same space, and we want to do an orientation. So what I think we can do is something that involves quaternions, but I'm going to try to avoid the quaternions and just try to muddle along. Man, it's been a while since I did this. But I have to do it, like, I have to do something involving quaternions and rotations and for whatever. Matrices. I have to do them frequently enough that I feel like I just need to like sit down and just really understand it. And every now and again I'll be like, yeah, I kind of understand what's going on here. And then very quickly I'll be like, no, I don't know. So I've got a few tools that I made for myself, like bond rotations and double bonds that do this kind of thing. Like, for example, actually, this is a good point. Oh, we should totally do this for each one. And I wonder if this is actually going to work. Because if each amino acid has the same point order for C and O, so this is the carboxylic, because there should be a double bond between the carbon and the oxygen in the carboxyl group. Wait, why is there only one? And the other thing that we're going to have to do is add water to each amino acid. Because if you think about it, if you're familiar with a little bit of biochemistry, when you have amino acids joined together, that process of joining them together, you remove 2 hydrogens and an oxygen from the molecule. So you have water left over, and that's why it's called a residue, because it's left over after the water leaves. But when we imported our valine, for example, here, let's just show this. Yeah. So we've got oxygen, and hydrogen. And on this end, we've got nitrogen and hydrogen. Now compare that with this guy. Let's hide this for clarity. We only have 1 oxygen on our amino acid, and we've got 1 hydrogen on our nitrogen. So we need to add 1 hydrogen there and a hydrogen and an oxygen there. That shouldn't be too hard, because if all of the amino acids are oriented consistently, then I can just add them to each of them. Just place them and add them one time. Just basically grab them from the valine reference that I have. And that shouldn't be too hard. So, but I need to make sure that they're all oriented the same way. So in terms of adding the double bond, well, let's just do that now. We can always change the points order later on. I was thinking through like a few different problems that I'm anticipating coming up, because that's part of the process. All right, these double bonds are way too chunky. Why? Oh. Wait, what? Why did this get... Oh, that's interesting. We have to add the double bonds after. Okay. Well, we'll not worry about that right now then. But I think in here, yeah, this is what I was looking for. This is what got me started on this whole thing here. Basically what I've got is some Vex code that does kind of what I want to do. I think. Let me just check here. There's also one that's tantalizingly called rotate and orient in here. And that's also what I did. And in fact, I think that maybe... yeah, I think that's what I'm going to use for now. Because I could certainly muddle through this and spend a lot of time Googling stuff and looking things up. But that wouldn't be very interesting for you, probably. Water break. WhatsApp break. My kids are outside. It's a beautiful day. How's the weather where you're at? Is it still feeling like winter, or is it feeling like summer? Or is it feeling appropriately like early April? All right. I don't know if I have enough brain power left in me for this rotate and orient. If that's the case, we might just pivot to so I'll try this for a little bit, and then I might pivot to doing some other things like rendering and laying them out on the grid. Should be pretty straightforward and famous last words, but maybe some rendering. I do recognize that I'm in a privileged position to have some outdoor play space. Not everybody can really get outside, which is in a time like this, which is hard, I recognize. But yeah, if you get some summery weather and you're able to get outside without getting too close to other people, that's really nice. We've had a few good days recently, which is great. Okay, so this rotate code. So what we want to do, let's get rid of that. What we want to do is we want to get two positions. I wonder if I should just use this as a reference and just kind of roll my own, because what we want to do. So I'm thinking to use dihedral. So what dihedral does is it computes the rotation matrix or quaternion, which rotates the vector A onto the vector B. Okay, what does that mean? Well, in basically what we have are, starting from this origin, which is that's what I want to do that first, have the alpha carbon at the origin. And then we've got a series of vectors, which are basically the angles at which we come out to the other atoms around it. So if we consistently say, okay, we take the vector from the alpha carbon to the other carbon, the carboxyl carbon, and rotate that such that we get the vector B, which is, it's hard to see. That vector, then we should be halfway there, because then we also have to orient the, what we could do is at that point, then just rotate everything such that rotate about that vector such that the R group is facing down. That kind of makes sense? maybe. Probably not, but we'll give it a go anyways. So first thing we want to do is we want to define our reference vector, which we will do by bringing in second point. So, okay, let's grab this stuff. I just worry that this part's going to be a little bit slow and technical to be very interesting, especially in my fumbling through it. But, you know, what time is it? 3:30. Yeah, we've been going for an hour and a half, something like that. I might... Yeah, you know what? Maybe I'll save this for another time. If people want, I can save this for another live stream, or what's maybe more likely is I'll just kind of figure it out on my own. But yeah, so then we won't be able to add the hydrogens or the water, and then we won't be able to add the double bonds. We could add the double bonds. All right, Dorothy and Wei J, thanks for joining me. It's great to have people here. It's nice to have. So interestingly, this process by which I'm talking aloud a problem that I'm having, and that's called rubber ducking in the programming field, because it's the idea that you have a little rubber duck at your desk and you say the problem out loud to the duck, and that helps you think through the problem. So I was starting to get a handle on the problem by just talking through it out loud, which is really helpful. But then I decided, you know what, I think maybe I'll just move on to other things. All right, so what we want to do, let's see where we can just quickly throw these onto a grid. So I've got a camera. Is that my camera? Yeah. So how do we want to arrange these? I'm thinking 20 divided by 4 is 5. And that's really the only evenly divisible way to do it. So I think that's what I'm going to do. And if it doesn't work out, then we'll try something else. Let's do 5 by 4. Is that right? No, I had it right the first time. And let's scale this out. So the idea being, we've got a whole bunch of points upon which we can throw our amino acids. This add node is really great because it's the best way to delete geometry. Naming for the win. All right, so we've got 20 different points, and we're gonna just do copy to points. So let's throw this over here. All right, so now we've got our alanine on each point. But the problem is, now if I turn off single pass, now we've got every single amino acid on every single point, and that's not what we want. We want, we want 1 on each point. So I think what I'm going to do, this may work, this may not. I wonder about sorting it. Sorting that. How would you sort amino acids? I was thinking to do sorting like in a biochemistry textbook where you have like, you start with glycine, right? Like that's the simplest amino acid. So you want to have that one in the top left. And then you kind of go over larger amino acids, and then you kind of finish with the aromatic ones, or I don't know, charged ones or something like that. I don't think I'm going to worry about that too much right now. I might tweak that a little bit more offline. But for now, I'm just going to leave them alphabetical because that's a sensible way to arrange them as well. So I did this before, so I'm going to, yeah, we gotta put this afterwards. There we go. So we're going to do this again. So pretty much the same loop. Whenever I say pretty much the same loop to myself, then I'm like, well, why aren't you putting it in the same loop then? Because I thought that I had to put the PDB structure after it, in between it, and I don't think I do. So what we want is we want a meta import node, and this is going to keep track of the iteration number. So this is like what index are we on for putting the amino acids on. I realize that these are kind of bunched together. We're gonna fix that. So let's do what's some good spacing. Something like that. I mean, for the big amino acids, that's not nearly going to be enough, but we might be able to stagger them later on. So, but for now, I'm just going to pull back like that. All right. So we've got our amino acids arranged, and we don't want to copy. We want to copy. I did this recently. I can't remember what I was doing it recently for. But basically, I want to copy the same. Basically, we want to get rid of everything that's not a single point. Is that what I do? Yeah. Whoops. So this is going to be. Is this the right thing? The latency is not too bad, actually, because I can hear myself talking over there very soon after I finish talking over here. Yeah, I think. No. I don't know. Is this going to work? We'll see. For each begin metadata. Whoops. I could probably do this in a, you know what? Yeah, I'm just going to do this in a, well, let's try this first. If this doesn't work, I have a backup plan. Always have a backup plan. So the iteration, I think it's just that. Ah, -1. Point index is not a valid group because I set it up to be primitives points. Okay. Whoa. Okay, that's not what I wanted. So wait, what? Okay. So basically what I'm trying to do, maybe I don't. Right. Point num equals. You know what? I'm gonna do a little bit differently. So point index. Oh, you know what? Forget that. I don't even need that. I think that is what I did before. So, minus one detail. Metadata node has a detail attribute called the iteration. And what I'm looking for is I want to get the index. No, I don't need -1, do I? Look at that. Look at that. All right, that's cool. So what I was doing there, without explaining what I was doing very well, is basically I've got the grid here, right? So I've got the grid and I've got 20 points starting from 0. You always start counting from 0 up to 19. And what I wanted to do is for each iteration of the loop, you get rid of every single point on that grid that isn't the same as the index or the same as the iteration number, basically the amino acid that you're on. So for the third amino acid, we only want the third point and get rid of all the others. There's probably a more elegant way to do that, but yeah, it works. So again, so what have we got here? We've got all of the 20 amino acids. They're arranged in a grid, and they're sorted alphabetically, I think, by 3-letter acronym, because that's how they're sorted. So what that means is we can always just kind of like tweak, right? Because this is procedural, right? I didn't lay them out behind, because that would be the alternatives, right? It's like how do you want the amino acids to be placed? You could place them by hand, but then if you're like, I don't want to sort them that way, I want to sort them a different way, you kind of have to do all that manual work again. And I don't mind manual work, but what I really don't like is doing manual work multiple times. So that's why I like Houdini's procedural nature, where I can just, get it in a procedural way, and then I can just tweak everything to my heart's content. All right, so obviously, so what are we missing? So we're missing the orientation. They're all oriented randomly, which is something that I can figure it out, but I don't really want to do that on camera at the moment because it tests my mathematics brain a little bit too much at the moment. We needed to add the water molecules to each, because as you can see, each of these amino acids should have... why does this one only have 0 hydrogens? Okay, so maybe that one will need 2 hydrogens, but the other nitrogens, I kind of want another hydrogen on there, and then we need another oxygen and hydrogen on there. And we do want to put the double bond between this carbon and oxygen, but again, I think I might leave that for off stream. What I do need to do now, so let's try to render this and maybe wrap up with the rendering, or maybe let's see. Don't have my... Oh yeah, I do have my time. Maybe do a little bit of comping, depending on if people want to hang around. I don't know if you yeah. See if people want to see a little bit of comping in Fusion. But we'll see how far we get. All right, so first thing we want to do, so I have this set up to have 2 different types of objects. What I want to do is I want to put the bonds, the sticks basically between the atoms in a separate object, so I can render them separately, so I can composite them separately in case I want to tweak them, which I won't, but it is. Yeah. So I'll put down a split. So I'll split out the atom prims. See, now you can see I just have the balls there. And the rest of it should be bonds. Indeed, that's the case. And so what that means is in here, we want to merge. Oh, it's already set up for us. How lovely. That's why the template is handy. So I also have a little tool to set up an environment for myself so that I can get a consistent look. So what I'm going to do is I'm going to drop my, you know what? I'm going to adjust my camera first. Because what I want to have is a little bit of room at the bottom for text. And I recognize that this is not going to be the final layout because these amino acids are not going to be in these positions. But yeah, for now, we'll just say that that's decent. So let's scale that up. And I'm going to drop it down a little bit, I think. All right. So I'm using Redshift for rendering, and hopefully this doesn't tax my, oh, it should be okay. Yeah. So I've got 2 GPUs for rendering, which is nice because that means that I can use one for stuff and the other for other stuff. All right, ta-da, we're done. There was a lot of look dev involved there, but I think we pretty much have what we want there. I'm just noticing there's like some interesting I don't think these are connections between molecules. I think this is just an artifact of stuff being in the same place. It just makes it look like it's let's see here. What we can do is we can turn on a single pass and kind of just see these one by one. Yeah, so it's that one that's merging with that one in a weird way. And that just has to do with the when I'm figuring out where the bonds need to go. It just uses like a distance. So we've got that one that's being weird and the other one's being weird. So what we could do is we could just make our grid a little bit wider, which won't help obviously because they're stacked. But we can make it a little taller. Not that direction. Nah. There you go. I'm thinking in my final layout for this shot, I think I'm going to have to do like a staggered. In fact, yeah. What if we did a little something like this? Those points, and we're going to okay. So what we want to do? Yeah. Maybe a little bit taller. Yeah, we're kind of running into, but anyways, this is not the final layout, so it doesn't matter. Am I happy with the RTX card? Yeah, I think it's a good card. So it's a RTX 2070. It's the current generation, so I'm happy to have it over the previous generation because it's faster than the 1070 for this sort of price point. Do I use the RTX optics functionality of it? No. I could try it and see if that helps, but the stuff that I'm rendering tends not to be too the kind of thing that you would really benefit from the RTX capabilities. But as a general card, I'm pretty happy with the 2070. It's so the 970 is the other one that I have that came out in 2014, and so I just like haven't. I didn't want to throw it out because it's still decent enough, but the 2070 is about twice as fast. It tends to render things about twice as fast as the 970. So I'm looking forward to new cards coming out, but more importantly, it would be nice to have Patreon support. I don't know. How do you ask for money? No, yeah. If you want to send me a better graphics card, no, I'm not gonna really pitch anything at this point. I would like to update my 970 card. It is getting a little old. It doesn't have that much video RAM. It's 3.5 gigs because, yeah. So it tends to go out of core if I'm doing a very complex scene, and having more video RAM is really nice. So the 2070 has 8 gigs. Although it only says it's got 5 gigs free here because it's being used for other things, presumably. All right, what was I doing? Do we still have this thing? Yeah, that's a mess there, but you know what? I'm not gonna fix it right now because that'll be fixed when I orient all of the amino acids properly. But for right now, that's fine. So what I've got, a background, and right now I've just turned off the primary visibility. But if I turn that back on, and I render this, then you can see that I've got a background there. But I'm gonna add the background in compositing. So I'm gonna turn that off. I was kind of didn't get to the animation part of the shot either. I think that maybe that's something I could do a different day. Yeah, you know what? I might try to like solve some of the more sort of onerous technical issues with this one, and then kind of save some fun stuff from this shot for another live stream. If people are interested in watching more live streams, if this was in any way educational or interesting or entertaining, or yeah, I think I'm gonna try and wrap it up in shortly after 4. Yeah. All right, so what are we doing here? So I've already set up my output, all my settings. I don't need to go into all of those. I've got, oh, actually maybe one thing so you can actually see the point of separating out the atoms and the bonds. So then I've got this puzzle mat here, which is basically color, red, green, and blue color identifiers for different parts of the image. And that way in compositing, I can isolate the bonds as separate things and the atoms as separate things. Again, I can't remember if that's actually something that I want to do, but it doesn't hurt to have, especially if it doesn't add too much render time. And considering that this, you know what? Considering that this shot doesn't take long to render, that puzzle mat is just fine. Here we go, the final render. Start your watches. Man, that's hilarious. It's just like this weird tangle of stuff. I like molecules. And it's done, 11 seconds. Which is, when we're doing animation, it's really good to have a low render time, because each frame, 24 frames per second. It adds up. All right, so hello again. Let's go find... Let's go find our rendered frame. Where did I save it to? It's a good question. I saved it to the wrong shot folder. All right. Thanks for joining us, Kevin. Appreciate you tuning in. Yeah. I'll see you. See you later. There we go. Okay, so there we go. So there's our beauty. Not too much to look at, I know. Do I not have a lot count view? I think that's how it's supposed to be. And here's our background, and let's stick them together. There we go. And let's put some text in. This text is going to say, what did I have in my amino acids? It's good text. And for consistency, so this is helpful to know for myself that that's kind of where I want my text, and that's kind of how big I want my text, which is maybe a little bit big for strictly necessary, but this is kind of a big like title, the amino acids. And so I don't want stuff to go below that. So I'm probably just looking at sort of the size of these. I'm probably going to have to back out, like just kind of back up a little bit and allow a little more space for this, unless I want to get really creative with the arrangement, because I don't want to have big like aromatic rings sticking down over here. Because in this particular shot, what I wanted to do was have some background boxes. And I know those people in the graphic design area will be like, no, don't put boxes behind each individual amino acid. But for the storytelling reason, I kind of have to do that. So let's choose a color. Let's make it the color of... I don't know. I think I've got a palette somewhere. So this is Fusion, and this is where I do my compositing. And if you've used a node-based compositor like Nuclear Fusion, then you know what's going on. If you haven't used any compositing software, or you've used After Effects, then this might look a little bit strange to you. But it's not too different from something like After Effects. Basically what we've got is we've got input media, like our background and our beauty pass. And then we just merge them together. So this is a merge node, and we merge them together, and you do stuff with it. So you change like the color space, and then you have some text, which, why is that weird color? And then you merge it in, and then you save it somewhere. And I don't want to save it there. Sequence 4, shot 2, 2A. Yeah, and so with my animations, I tend to be as minimal as I can with the compositing, just because there's often so many shots that I can't take too much time to be tweaking every aspect of every individual shot. So I tend to have kind of like one consistent sort of look, or a few different looks depending on what's in the animation, and try to get most of it looking the way I want to in the render, in Redshift render. So that's why I'm not doing too much color correction. So this is, and then I bring in the puzzle mat if I wanted to. So yeah, so here's our puzzle mat, and then we can split it out into the red channel, the green channel, and then put that into the alpha, and then we can use that as a, or yeah, bring them together. So if I wanted, for example, let's say I wanted all of the bonds to be tinted blue. Ta-da! See, all the bonds are tinted blue, or purple, or red. Maybe I wanted to like kind of boost them a bit, make them like really like, oh, they're glowing. These bonds are red hot. Yeah, that's the benefit of that. But as I said, I'm usually decently happy with them as it comes out of the render. Sometimes a little bit of a tweak here and there. And sometimes there's effects that you can only do with compositing, or that would be very difficult to do in render. Often if I'm adding depth of field, blur, and stuff like that, then I'll want to, speaking of which, I don't think I outputted a depth pass, but that's okay. All right. Yeah, so kind of one of the things that I wanted to do was kind of put some boxes behind these guys here. And this is one thing that I haven't really figured out a great way to do, because each of these is a different size. Like one thing that I considered doing was if I duplicate the input and then offset them like so, you know, I could do something like that, and then something like that, but that's not really going to work so well for the individual sizes. So I feel like this might be something I'm going to have to do manually. So I don't really want to spend the time to, you know, arrange that until I've actually got the final layout and the size of all the amino acids and everything like that. But that's kind of the final sort of shot and layout as part of the animation. So when that comes out, you've got to be introduced to all these amino acids, and you'll have a behind the scenes look at how that came to be. Yeah, so I think I'm probably going to wrap up the live stream here. The couple of things. Yeah, okay, so I said I wasn't going to do any promotional stuff, but I am. So this is what you're watching right now on Making Biocinematics. And right now I've got 84 subscribers. You know, it is what it is. But what I need in order to create a YouTube URL that says youtube.com/makingbiocinematics, so I can more easily point people to live streams, etc. If you could go ahead and click this subscribe button, that would be fantabulous, because then when I get to 100 subscribers, I can have that nice vanity URL. And you know I'm all about the vanity. Yeah, but this is the channel where you can see behind the scenes for biocinematic stuff. I did a presentation. There's a behind the scenes for a diffusion video. It came out in the fall, far too long ago. But yeah, so now I'm, yeah. And if you're completely new to this channel, this is the main channel, Biocinematics, and this is where I make most of the videos about molecular animations. And one of the ones this is the trailer for the one that I was working on right now. Hopefully that'll come out soon. And in the future, I'll be able to share some client work that I was working on in a new sort of format, which I'm excited about. And we'll see, but I don't, because that research hasn't been published yet, I can't post it. So I kind of have to wait for that to happen. And that may take an indeterminate amount of time. But at that point, I'll be able to share an awesome animation about some research. And what else did I want to say? So if you want to subscribe to Biocinematics, that's really super helpful for me. And if you are really keen on seeing this project continue on, one thing that is super helpful, well, beyond just like sharing the videos and watching them and all of that good stuff, is I have a Patreon page. There are a number of like 9 different tiers that go from $1 per video. And as you can see, I'm not putting out a video very frequently, so you're going to be paying very little in terms of the grand scheme of things. But I also understand that a lot of people are under more financial pressures at this time in history, and that's completely understandable. So I'm not putting any pressure on, but if you're interested in that kind of thing, we've got $1 per video, and then there's some perks for other things. Once we get to 100 patrons, you'd get exclusive access to live production streams like the one that you're watching right now. So right now I'm having these open to the public, but in the future there may be additional live streams that are for patrons only, or maybe most of the production streams will be exclusive to patrons only. I'm not quite sure yet, but that's something for the future. And then there's other things you can check it out. So I hope you'll at least take a look at it and see if there's anything interesting for you. One goal that we reached in the recent past was to get a new webcam. I don't have a new webcam yet because I actually haven't put out a video since this, like a main video, since this video, since the goal was reached. But I will be buying a webcam after that point. And so hopefully the quality is going to be a little bit, it's kind of flickering. It's not too bad, but I feel like this kind of like fuzziness and color quality kind of bothers me from a making videos standpoint. Anyways, but yeah, so there's other goals. And there's posts, there's wallpapers and project updates and stuff like that. So if you're interested in more of the behind the scenes stuff, you can maybe take a look at that. And that's pretty much it. I think I'm going to wrap up here. Thank you so much, everybody, for joining me in this inaugural production live stream. I had fun doing it. I hope you had fun watching it, and maybe we'll do it again soon. If you have suggestions on what, like, times of day that work best for you, I know that people are in various time zones, but let me know what kinds of things work best for you. If this is the middle of the night for you and you're just would rather it was in the morning or the afternoon or whatever. Anyways, yeah. So I'll keep you posted on Twitter. Twitter at Biocinematics on Twitter. Follow me, and for updates, Instagram, whatever. You can figure out where I am with social media stuff. There we go. So yeah, thanks again. I really do appreciate you tuning in, and hope to see you next time. All right, take care. Stay healthy. Stay safe. All that good stuff.